Our FTIR microscope is a tremendous resource to investigate potting quality, potting anomalies, or potting contamination. First, FTIR stands for Fourier Transform Infrared and is an infrared spectra absorbance of the chemistry of a substance. The infrared spectrum absorbance of a substance is a sort of chemical fingerprint that yields information about the chemistry of that substance. For example, in this spectra, you can see the broad absorbance at approximately 3,500 inverse centimeters, which is characteristic of a hydroxyl peak of a polyol, or the sharp peak at approximately 3,000 inverse centimeters, which is characteristic of a methylene group of a plasticizer. Because molecules are made up of several different functional groups, the absorbance pattern of a specific molecule will be unique to that specific molecule. This is also true for mixtures of molecules, where the mixture will have a characteristic FTIR absorbance pattern based on the functional groups it contains. There are two primary methods that we utilize at Epic Resins to measure the infrared spectrum absorbance of a substance: ATR and ATR microscope. ATR stands for Attenuated Total Reflectance. And ATR works by placing the substance you intend to analyze against a crystal energized by an IR laser. When an evanescence wave is produced by the laser light, it comes into contact with the sample and absorbs some of the IR energy, yielding a single absorbance spectrum. This spectrum is specific to the chemical functionality that came in contact with the ATR crystal. An ATR microscope uses the same crystal types and technology as a standard ATR FTIR, but instead of producing a single IR absorbance spectrum, it measures the IR spectrum absorbance on the surface in an array. This feature allows us to detect chemical differences on a surface. This is very useful when you need to determine the chemical variation of a surface. To demonstrate the utility of the ATR microscope, we have two projects to share with you today. The first is an electronic application where the customer was concerned that there may be variation in the polymer at different portions around a sensitive component in their application. We started the analysis by sectioning the parts to the desired region where the sensitive component contacts the potting compound. As can be seen in this image, the tan portion in the center is the sensitive component. The gray area around this is the potting compound. The customer was concerned with the regions marked 1 and 2 and wanted to determine if there was a difference in the potting compound in these areas. We analyzed a 100 by 100 micron square area in the regions labeled 1 and 2. These FTIR microscopic images show the array of the FTIR absorbance at these two regions. In looking at these microscopic images, we extracted the actual IR spectrum absorbance from these different regions. These are the IR absorbance spectra for regions 1 and 2. When we overlay the two IR spectra and measure the statistical correlation between the peaks, we find a 98% correlation between region 1 and region 2, indicating a high probability and minimal difference in the chemical functionality of the two regions. The second demonstration of the utility of the ATR microscope was an instance where a customer was observing inconsistency on the surface of their potting material at the completion of their curing process. The image here shows what the customer was observing. We again started this analysis by sectioning the potting compound in the suspect areas. Upon analysis, the following ATR microscope absorbance pattern was produced. You will probably notice the significant color variation in this example compared to the first example. This is because the ATR microscope uses color variation to visually show where there are significant variations in the FTIR spectrum. If we look at the spectrum of the pink areas, you can see at 2275 inverse centimeters, the absorbance is minimal. However, the spectrum of the blue dots show a distinct peak in this region. To compare these regions, we superimposed the spectrum absorbance of the blue dots onto the spectrum absorbance of the pink areas. You can see a distinctly different peak at 2275 inverse centimeters. This is a NCO functional group. NCO is the reactive component of the B side of this formula, and its presence in the FTIR shows that it is unreacted because it was not properly homogenized during the dispense process. In looking back at the FTIR array, the dots that are blue in the array are areas where the B side was not sufficiently mixed, and the pink regions are areas where sufficient mix was achieved. Upon sharing these results with the customer, they investigated their meter mix and dispense equipment and found that they had a partially plugged dispense head manifold, which was causing a lead lag condition during the dispensing shot. After completing necessary maintenance protocol to their equipment, we were able to re-verify their mixing process was correct. These are just two examples of the utility of the FTIR microscope. We have also assisted customers in identifying surface contamination, thermal degradation, excessive mold release, counterfeit thermoplastic case material, and many other proprietary investigations. The FTIR microscope is an indispensable tool to investigate post-potting anomalies.